In this lecture we're going to talk about the control of muscles. We're going to talk about how muscles contract, the size principle that determines how we recruit muscles in an orderly fashion. We'll review some basic central pattern generators, and then we'll talk about the sensory and cortical inputs that control our somatic efferents. So the innervation of the muscles is going to occur at neuromuscular junctions. This is where motor neurons are going to release acetylcholine to cause muscle contraction. Now the basic building blocks of muscles are sarcomeres, where we have our thin actin filaments held together at Z bands. And in between those we have our thicker myosin filaments. The sarcomere is going to change its length by sliding these filaments along one another. Myosin has the motor proteins. Actin is going to have binding sites for myosin that will get exposed in a calcium-dependent manner. As these actin filaments slide toward the center due to the movement of myosin, our muscles shorten and contract. So the sarcomeres are the basic building blocks, and they're made of actin, that cytoskeletal filament, and myosin, the motor protein. These sarcomeres are going to then create our myofibrils, and the myofibrils will come together to make a muscle fiber. But the sarcomeres are the basic functional units of muscles. So they're going to actually generate tension through the activity
is
of
triggered by a series of molecular events
proteins
known as the cross-bridge cycle.
Forgot to mute that. So what we're going to see here is that input from motor neurons is going to cause the shortening of muscles. And this happens because of the sliding of actin along myosin. So little myosin heads are going to pull on actin and make this shorten. This is going to be carried out through ATP hydrolysis of the myosin head. It's going to allow it to carry out that power stroke. So myosin can really only hold on to two things. Either it's going to hold on to that adenine nucleotide, whether it's ATP, ADP, or it's going to hold on to myosin. Or it's going to hold on to actin. So myosin can only hold a certain number of things. One. So imagine it's your hand. It can only really hold one thing. So myosin's preference here, first, it wants to bind ATP. After that, it's okay with actin. And the last thing that it wants to bind is ADP. So it's always going to be hydrolyzing ATP to form ADP. That hydrolysis there is going to move the myosin head forward. When the myosin head is forward, that's going to allow it to then bind to actin, because we've hydrolyzed ATP to form ADP. So what it'll do is kick out the ADP and grab onto actin. When bound to nothing here, that's going to cause the myosin head to flop backwards. So there's going to be that power stroke. And that's going to pull the actin filament about 10 nanometers or so toward the center. That's going to shorten our sarcomeres, and that's what's going to allow for muscle contraction. So here's a little movie showing how this is going to work. So when myosin binds to actin, again, it can only hold on to one thing. It would rather hold on to actin than ADP. So we're going to release a phosphate. We're going to release the ADP after we've securely bound to actin. The release of ADP is going to cause myosin to then cock back and pull on actin. Of course, this whole cycle will be reset whenever a fresh ATP bumps into the ATP binding site. And again, the preference is going to be ATP over actin. So when we bind ATP, we're going to have to let actin go. So ATP binding is going to destabilize interactions with actin. So we're back at our first preference, but of course myosin is an ATPase. It's going to hydrolyze ATP to create ADP. and then I'll push it back forward. And then we're back where we started. Now, whether or not actin and myosin interact with each other depends on the amount of calcium floating around. What calcium is going to do is bind to the calcium binding protein troponin, and then troponin is going to pull tropomyosin out of the way. When calcium levels are low, tropomyosin obscures myosin binding sites on actin, so myosin can't stick. What nerve cells are going to do is allow for an increase in intracellular calcium within the muscle. That increase in calcium is going to then cause troponin to pull tropomyosin out of the way. Once tropomyosin is out of the way, then we can get our actin pulling from myosin, because myosin can now finally bind. So the function of nerve cells is to provide the excitation needed to increase calcium levels. What we're going to do is act on nicotinic acetylcholine receptors to increase calcium levels. Nicotinic acetylcholine receptors are a little bit calcium permeable, but more importantly, they depolarize the muscle so that it fires an action potential. That action potential then spreads throughout the muscle and causes the release of calcium from the sarcoplasmic reticulum. So here, if we have a look, particularly in part B, the blue line is showing you an increase in calcium. The green line is showing you the length of muscle. Notice as calcium increases, the muscle length decreases. And that's because of the actin-myosin interactions pulling on the sarcomeres. So in the cartoon world, what happens is the release of acetylcholine from a motor neuron. Acetylcholine stimulates nicotinic acetylcholine receptors. The muscle cell fires an action potential. This stimulates voltage-gated calcium channels, which then allow for calcium release from the sarcoplasmic reticulum through ryanodine receptors. Once we have an increase in calcium, troponin pulls tropomyosin out of the way, and we're off to the races. And our muscles shorten. Now, the neuromuscular junction is a very strong synapse because it has a large number of active sites, about 1,000 of those, and it has those post-junctional folds that are going to trap acetylcholine, just like they did in previous lectures. So we're going to have a very large, very strong synapse. Now, the order in which we recruit motor neurons depends on their size. So we're going to follow something called the size principle so that we have orderly recruitment of muscle fibers, depending on their type. There's really two broad types. There's slow twitch, fast twitch. And in fast twitch, we have fast fatigable and then fatigue resistant. The type 1 fibers are going to be the weakest, but they're going to be able to work all day, so that you find a lot more of these in postural and respiratory muscles, because you don't want to get too tired to breathe. Then it's over. So what we see is an increase in the amount of slow twitch in our respiratory fibers as we age and we use those muscles all day, every day. And the muscles that we use to generate a large amount of force, but a little infrequently, those are going to be more enriched in the fast twitch muscle fibers. So type 2 fibers here. Type 1, slow twitch, they'll work all day. Type 2, fast twitch. Slow twitch is going to use primarily just aerobic metabolism and generate a lot of ATP while expending very little force. The type 2 fibers are going to use more anaerobic metabolism and generate a whole lot more force. So you can see here the 3 different types of muscle fibers based on staining for different types of myosin ATPase. And then looking at glycolytic enzymes and the electron carriers that we use in oxidative metabolism. So you'll see a lot more glycolytic enzymes in your type 2 muscle fibers, and you'll see more NADH in your type 1 fibers. Now, based on usage, you can change between these different types of muscle fibers. So what they did was actually cause a conversion towards slow twitch fibers by very weakly stimulating the gastroc in cats. So they put in electrical stimulators, they stimulated the nerve constantly, but at very low amplitude. Notice the change in force generation in that curve. We're going from a whole lot of force very quickly, this is baseline, to that lower levels of force. It takes a little bit longer because it's a slower contraction. What this is telling us is now we have our slow twitch far more abundant than they were originally. There was some there, but we increased them through use. So our muscles are going to change their makeup based on how much we use them. Now the recruitment of these different muscle types depends on which motor neurons we're stimulating. So just like we have the three different types of muscle fibers, we also have three broad classes of alpha lower motor neurons. Type S is going to innervate slow twitch fibers. These different types are of different sizes. Small, medium, and large, just to kind of lie to you a little bit. Type S are the smallest. That's type S. Then we have our fatigue resistant and our fast fatigable. So this is the size of the motor neuron. This is the motor neuron. The muscle fiber types that it hits would be type 1, 2A, 2B. Most force, least force. So we're going to get orderly recruitment here because of the size principle, which is just a restatement of Ohm's law, of course. So the smaller ones have much higher resistance. Therefore, you're going to get a larger potential from the same current coming in. They're going to be recruited first, so that whenever we begin we begin a movement, we recruit the weakest muscles first and then add on the stronger ones as needed. That's what we see in the size principle. So we're going to start with the weakest, and then we're going to add additional muscle fibers as needed. And that's what they're seeing here. So they have to computationally tease out different types of motor units from their EMG recording. But they found about 5 different units that were recruited in a fairly orderly fashion. The weakest one, A, recruited before B, before C, before D, and before E. And those correspond to larger motor neurons and stronger muscle fibers. So again, it's just Ohm's law. That's it. The smaller the motor neuron, the easier it is to reach threshold because it has higher resistance, so charge builds up. The larger the motor neuron, the more current you need to hit threshold. So first we get the small weak ones, then the big strong ones. Now in the spinal cord, we have inhibitory neurons called RENSHAW cells. Hold on a second. My camera is not happy. I'm going to edit this out. Okay, cool. Now, in the spinal cord, we have more than just motor neurons there. There are, of course, inner neurons that are going to control the activity of these lower motor neurons. And one of them there is the RENSHAW cell. This is showing you an immunofluorescence stain for RENSHAW cells. So they are indeed glycinergic, so these are inhibitory neurons. And what they're going to do is provide feedback onto motor neurons to better pick out which ones are being stimulated. So whenever we have corticospinal tract input down in the vague area of our lower motor neurons, RENSHAW cells will help specify this input. So we have kind of diffuse input from the corticospinal tract onto lower motor neurons, some of which will be stimulated, let's say very, very strongly, and others not so much. We got to remember there's always some slop in our axonal projections. What RENSHAW cells do is provide some negative feedback. So when a motor neuron is stimulated, that stimulates RENSHAW cells. It's also going to go act on the muscle, of course. And those RENSHAW cells are going to quiet down the neighbors. I've shown you here in this cartoon. So in this case, we're getting strong input to motor neuron 1. It has an increase in activity. Motor neuron 2 has a decrease in activity. So we're better teasing apart the pattern of lower motor neurons because of the activity of RENSHAW cells. Now the movements that we carried out are going to be generated by pattern generators, or collections of inner neurons that help us create a consistent pattern of output. Because truth be told, we don't give a lot of thought to most of our movements, and a lot of it's carried out non-consciously. We have central pattern generators in our spinal cord and in our brainstem. They're going to help us create movement. So all we have to do is think, I want to walk, and they work out all the details of putting one leg in front of the other, slightly leaning forward to move our center of balance. If there's any little deviation in the ground, they'll pick that up and make sure we don't fall over, and we give it very little thought. All we have to think of is walk. They then work out the pattern. Now, a very simple pattern generator to see would be that of the stretch reflex. You tap on a tendon and you get a kick. The central pattern generator here is just a single interneuron. It's really simple. So we got our muscles here. We got a knee that looks nothing like this. When you tap the tendon, this is going to stretch the muscle. That stretch is of course going to be sensed by the muscle spindle, like we talked about in lecture 16. The muscle spindle then provides input to the spinal cord here. We're going to hit a couple of targets. One's going to be an interneuron, and the other is going to be a lower motor neuron. So when we stretch the muscle, we stimulate the afferent. That stimulates the interneuron, which inhibits the antagonist lower motor neuron. So we relax this muscle, allowing it to lengthen, and we stimulate the agonist. So we contract this muscle. And what that leads to is a kick. Now, the stretch reflex is mostly there to cancel out noise, but it shows us this very simple pattern generator. When we're stimulating the agonist, we inhibit the antagonist at the same time. That interneuron is a very simple pattern generator. So when you try to move at a joint, you don't encounter resistance. Pretty straightforward. Others are slightly more complicated because they involve just a few more neurons, but what they allow for would be, let's say, alternating movements on one side of the body and the other. So if you're a snake, you slither. And if you're a human, you move one leg and then the other. You don't hop, you walk. So we have interneurons that inhibit the other side of the body. So when the right extensor is active, the left extensor is inhibited. Conversely, when we're kicking our left leg forward, that inhibits the right. So we're no longer putting the right forward. When we put the left forward, the only movement that we allow is to bring the right back. So we get alternating movements by having pattern generators inhibit one another. Pretty nice little system. And what that leads to is alternating movement. So those inhibitory commissural interneurons allow only one side to be active at the same time. So whenever we have excitation on one side of the spinal cord, that's going to lead to inhibition of the other. So we're down here in the anterior horn, and one of the nifty pattern generators that we have would be reciprocally connected excitatory interneurons. When this neuron's excited, it excites its partner, which excites the original, which excites its partner. So you can imagine this is going to lead to a burst of output. Now, of course, calcium-gated potassium channels lead to the pause. So after we burst and calcium builds up, that triggers potassium channels so that we can have a pause and let calcium drop back down. When these are active, they're going to stimulate lower motor neurons, so we get robust output and strong muscle contraction, and they'll also stimulate an interneuron that goes and inhibits the other side of the spinal cord, so that when we're moving one side, we don't move the other. Now, of course, we can turn these off, so we can move both sides at the same time. The end of the burst, of course, as I mentioned, is caused by calcium sensitive potassium channels. That's what we're seeing here. So in this case, they're just recording calcium levels when they cause bursting of these excitatory interneurons. That's it. So when you see a burst, you see an increase in calcium. That then stimulates the potassium channel, and we end the burst. Those inhibitory interneurons then coordinate both sides of the body. So a lot of our movement is actually on autopilot, believe it or not. And I think this is best illustrated by watching this classic movie here of a decerebrate cat. What they did was cut and remove the cortex. So they cut above the brainstem and the spinal cord. So we have midbrain intact, pons, medulla, spinal cord. But the cerebrum's all gone. So they have to support the weight of this cat, but I want you to notice that it's first walking, then it's jogging, and then it's running. It's recreating all the normal movements when they start the treadmill running. So here, slow speed, you get something that looks very much like a walk. As they ramp up the speed of the treadmill, it's going to go from a walk to a trot. And as they continue, it's going to go from a trot to a gallop. The cortex is all out of this. This is all just central pattern generators running on autopilot. Pretty nifty stuff. Now these central pattern generators normally are under the control of descending tracts from the cortex. We aren't decerebrate people, unlike those poor cats. Now the motor output is of course going to be regulated by several other regions of the central nervous system. There's the basal ganglia, which we'll talk about in the next lecture. The hypothalamus, as we've already gone over, that's going to help us regulate our behaviors. Are we thirsty, for example? Well, we should go find some water and give it a drink. The motor cortex itself has the lower motor neurons, and of course if you have damage there, you'll have problems with movement, but hell, you can get damage elsewhere and see issues with movement, such as damage to the cerebellum. So here we can see a patient with spinal cerebellar ataxia. So what they're doing is having her just, quickly as she can, go ahead and tap her fingers there. And that might have seemed a little weird, but it wasn't too bad. Now notice the left hand here. Quite a bit more difficulty, first of all, just coordinating the movement and then getting it done quickly. So the cerebellum is going to help with these multi-joint movements, like tapping your fingers together or putting your finger to your nose and then touching something else and back to your nose. So coordinating those movements all at once so that we're moving in a straight line and we're just touching our nose, not going too far. Notice there that little bit of miss, and then there's a little tremor at the end. Textbook cerebellar damage. So the issue that occurs here, the cerebellum is unable to communicate back to the cortex and tell it how to do what it wants to do. If you ever want to get an idea of what your cortex is like without your cerebellum involved, if you're right-handed, try to write left-handed. Your cerebellum doesn't really have it figured out like it does with your other hand there. So we have all kinds of motor programs dedicated to writing with the hand that we use a lot, but with the hand that we don't, it's all going to be cortically driven. And you can see how good of a job your motor cortex does when we don't have those motor memories in the cerebellum. Try to do it at the same speed as you'd write with your right hand. Looks like shit. That's because we don't really do a good job moving with our cortex. It provides some pretty vague plans. And the spinal cord and other central pattern generators figure it out. Now, of course, we have sensory input to help regulate our motor output. So we have that refined descending input from the corticospinal tracts after it's gone through the cerebellum and basal ganglia to be coordinated and approved. We're going to refine everything based on sensory feedback, just like that cat did. When the treadmill was moving faster, it could tell its limbs were moving faster, and so it changed the pattern of motor output to match that increase in speed. We can see the effect of sensory input by doing some simple reflex testing. Nothing too tricky there. The amount of autonomic activity will also shape the output of our motor pathways. So, the activity of motor neurons then is going to be affected by other neuromodulatory transmitters, such as things like serotonin and norepinephrine. Serotonin and norepinephrine are going to increase the activity of motor neurons by generating plateau potentials, so that they are much more excitable. And they'll prolong the output of motor neurons as well. They're going to do that by increasing intracellular calcium levels by acting on their metabotropic receptors. So you can see here the effect of applying BAPTA-AM. This is a calcium chelator. When we soak up all the calcium, the neuron is only active during the depolarizing current, whereas on top we deliver a depolarizing current, then we remove it, and the neuron continues to fire for some time. That's going to be because of the activity of the plateau potentials that are generated. We're going to get long-lived depolarization from neuromodulatory input. So this can allow more than just the corticospinal tract to affect motor output. That's all we have for this lecture. Please use the question box to let me know what isn't clear. I'll see you later.